I'm going to talk about one simple example, so-called CpG island prediction. The basic idea is, as you know, DNA has a four-letter alphabet, A, C, T, G, and it is linked from nucleotide to nucleotide by so-called phosphate groups, here written with the letter P. So CpG islands are regions of DNA sequence that are highly enriched for the dinucleotide C followed by a G. And the P just reminds you that it's linked by a phosphate group in between. This has biological significance in a couple of ways. One is structural. So in this picture, I think you can recognize the DNA double helix winding round and round itself, and then going around this central object, which is a representation of a so-called nucleosome, which is basically a complex of histone proteins. Basically, the job of nucleosomes is to, at the lowest level of structure, pack the DNA in a cellular nucleus into kind of convenient structures, both for the purposes of just structural packing and also for the purpose of regulation. Whether DNA is wrapped tightly around nucleosomes or opened up, not packed tightly around nucleosomes, figures importantly in control of things like gene expression. In other words, if a gene's promoter is tightly bound versus not, that can have a strong effect on basically the production of messenger RNAs from that gene. Long story short, CpG islands have strong enrichment of C followed by G dinucleotides. You may notice that in some sense this is a double-stranded palindrome. That is to say that the complementary sequence to this on the other strand is going to be what? CG. Right, we're going the other direction on the other strand. So it looks the same no matter which strand you're looking at it on. These highly enriched regions called CpG islands are themselves enriched for binding of specific proteins that regulate gene expression, and also for chemical modifications such as methylation, which then themselves, those chemical modification events, influence regulation of gene expression. This is even important in disease. For example, in cancer, CpG islands are involved in the shutdown of genes that act as tumor suppressors. So basically tumors, if you will, misuse CpG islands to selectively turn off genes that would normally kill off that tumor cell, and that escape from programmed cell death is actually one of the ways that a cell can go down the road to becoming unregulated growth, in other words, a tumor. Okay, so